So thank you, everybody, for coming, and welcome Dan Knight to the Cancer Center seminar. Appreciate you coming. So his expertise, as you'll see, is in computational biology. That is not my expertise, and I'm looking forward to hearing what he has to say about the microbiome and how it's impacted by chemotherapy, which is really one of the things he's been focused on. He's an assistant professor in the Department of Computer Science in the Biotechnology Institute. And prior to that, he completed a postdoctoral fellowship at the Broad Institute of Harvard and MIT. People know about that, certainly. I asked him what made him come here, and he said, well, it's a great place to do microbiome research. So that's terrific. Not a great place for football, but a microbiome research is terrific. That was a joke. It didn't work. Didn't work. So, you know, his interest is in developing algorithms to improve diagnosis of dysbiosis, which we'll hear about. And that's a condition in which the gut microbiome is out of balance due to high-dose chemotherapy. And it puts the patients on chemotherapy at an increased risk for several things, including systemic infections. And he'll maybe talk about that. And one of his goals is to use these methods to identify shifts in the gut microbiome, which could provide biomarkers and ultimately be used to help guide clinical practice. And as you know, the microbiome is becoming increasingly implicated in so many pathologies. It's just fascinating, in my view. And so I appreciate you coming, sharing your thoughts. And Dan, welcome to our seminar series. You're welcome.
Thank you so much. It's a pleasure to be here. We do computational biology in my lab. The talk today is going to be mostly focused on the biology part, so I won't be forcing you to eat your vegetables and talk about algorithms. I want to start with just disclosing a conflict of interest that I'm part of a company that's commercializing some parts of microbiome analysis, although everything I'll talk about today is open source and public. So dysbiosis is this kind of buzzword these days. It's kind of intentionally vague, but it basically means an imbalance in your gut microbiome, in the inner ecosystem. And there are many different types of dysbiosis, but the notion of having an unhealthy community of microbes inside you is actually it's important, and it's been linked to this growing list of major chronic, virtually every major chronic human disease has now been linked to a shift in your gut microbiome. So the microbiome is this world of microbes inside you. It's about 500 or so species in a given healthy person. There are more microbes inside you than there are human cells in your body. Of course, the human cells are bigger, so that's why you look like a person. And it's got this enormous amount of functional complexity. There are somewhere around 100 times as many genes, different types of genes, in your microbiome as there are in your own genome. So they're basically like these little chemical factories inside you, converting drugs, delivering drugs of different kinds to you. And we don't know what the vast majority of those genes are and what they're doing. But we know that if you have a shift in the microbiota in a certain direction, it can cause obesity. You can actually put microbes from an obese person into mice, and the mice will become obese. And we've done some work like that in my lab as well. It can contribute to diabetes, malnutrition, asthma and allergies, a number of autoimmune disorders like Crohn's disease and ulcerative colitis. And these are all interestingly modernized, kind of developed nation diseases. So we know that in modern countries like the USA, there's this depletion of the microbiome. People lose diversity when they come to the USA. Even immigrants, it happens to them. And at the same time, these diseases are all skyrocketing all over the world in developed nations. So there's sort of a smoking gun pointing at the microbiome. The microbiome modulates the immune system. This is a figure showing in each column a different strain of microbe, and each row a different type of cell. in a mouse. And you can see that the different microbes are inducing different gene expression patterns in these cells. So, there can be very specific effects depending on which microbe it is and which host cells are being exposed to it. And the microbiome determines drug metabolism. There's been a lot of exciting work in chemotherapeutic agents. The microbiome, in some cases, inactivates drugs. So, for example, digoxin, which is a heart disease drug, if you have a certain strain of Agarthella lenta, this is a microbe that many of us probably have, then it will inactivate digoxin and cause you to need a much higher dose. It also has been shown to reactivate drugs after they go through the liver, and that can cause dose-limiting toxicities in some types of chemo, because all of a sudden there's this big bolus of the drug that's being released in a localized setting inside the gut microbiome, inside the gut. So the microbiome is part of your drug metabolism. And I think there's a lot of exciting work to be done on dosing and personalization of drugs based on whether you have the pathways in your microbes to break down or reactivate a drug. And finally, what we'll be talking about a little bit more today is that the microbiome protects you from infection. And this has really been kind of the poster child of the microbiome field, that people who come down with a recurrent Clostridium difficile infection, which is extremely common these days, somewhere around 1% of all hospitalizations nationwide are now resulting in a C. diff infection. And then maybe 10 or 20% of those people, you give them a new antibiotic and the bug comes back. Then you give them another antibiotic and maybe 10% of people, it comes back again. And so those people have recurrent C. diff, and they're basically stuck being on antibiotics for the rest of their lives. They might have to have part of their colon removed. It can be fatal. And there was work done, actually the pioneering work was done here at the University of Minnesota by Alex Koretz and Mike Sadowski, showing that you can take a healthy person's fecal microbiome and transplant it into a patient with a C. diff infection, and it will cure them. Like within hours, they get up and walk out. And it's a 90 to 95% cure rate. This is something that was previously untreatable. And it's really kind of a sledgehammer approach. I mean, you're literally taking poop from a healthy person and transplanting it into the person with C. diff, and they get better. So there's been this shift in thinking about pathogenicity and infections as being not just a two-party system where it's pathogen versus host, instead to this pathogenicity in a complex ecosystem. So it's really the pathogen versus the host and the whole microbiome that's there. And this may actually be important in chemotherapy as well. So the C. diff case I mentioned, that's where you get antibiotics, and the antibiotics disrupt your microbiome, and the invasive species takes over. It turns out chemo does somewhat the same thing. So if you think of the microbiome as a healthy rainforest, this is your gut on chemotherapy. It literally causes almost as much disruption to the microbiome as antibiotics. And this is one of the things that we're studying in my lab, is characterizing different types of dysbiosis. This is a very extreme case. And understanding how the baseline microbiome impacts a person's chances of success and avoiding infection when they go through chemo. The way we do this is by sequencing the microbes. The vast majority of the microbes are hard to grow in the lab. Some of them actually need each other to grow. Some of them need to be in very certain chemical conditions to grow. So rather than growing them, we grind them up and shear the DNA and sequence it, and we get something that looks like this. So this is your gut on my supercomputer. And we literally, this is what we do. We line up all of the genes and we look for subtle variations that tell us How diverse is this microbiome? What species are there? What genes are there? And then when we do this for many people, we can begin to understand epidemiologically what are the risk factors for clinical outcomes. This is a special microbiome. This is the microbiome of a patient who's about to go into high-dose chemotherapy. And she actually goes on to, she makes it through with no bloodstream infection. And you can diagnose, or you can actually tell that from looking at this slide. So if you squint at it, can you tell that her microbiome is diverse? What you're looking for are subtle variations. So like here, this sequence came from one bacterial cell in her stool sample. The next sequence came from another one. And they're about the same, but you might see, okay, here's a difference. And if you walk along, there's another difference. And if you were to tally this up, there are actually about a dozen different species represented just in these first 25 or so sequences from her gut microbiome. So this is a patient who has very diverse microbiome and is going to survive the chemo. We started some work in this area with Emmanuel Montassier, who did a visiting postdoc in my lab and is at the University of Nantes in France, which is a big bone marrow transplant and stem cell transplant center in France. And we first wanted to find out, what does chemo do to the microbiome? And this is all 5FU. The effects were pretty stark. So this is a distance plot showing you 1 point for each stool sample. So this point is a whole zoo of microbes. And all it's trying to show you is the ones that are close to each other have more similar microbes in them. So from this, you can see the before samples are colored green, and they're over here on the right. And then the after samples from the same patients are colored red, which are all over here on the left with a couple mixed in here. So there was clearly this major shift following the chemo in these subjects. And so it's kind of a broad ecological shift. We also see a bloom of Proteobacteria. So these are the mixtures at the phylum level of different phyla in each patient. So each patient is a column here. And then these are patients before. You can see that they're dominated by this phylum Firmicutes. After, they get this massive bloom on average in Proteobacteria, which tend to be kind of inflammation-related microbe. And there are major functional changes as well. So we see increased xenobiotics degradation pathways, sulfur metabolism, which could be involved in production of hydrogen sulfide, which is a genotoxin, and has been associated with increased gut permeability, and flagellar assembly, which is kind of a virulence factor. So they're getting, it seems as though their microbiome is getting worse, whatever that means. It's more virulent. It could be producing more toxins. So there have been no outcomes here. It was just a pre-post chemo. And we were interested in examining bloodstream infections and sepsis. So bloodstream infections are actually very common in people getting high-dose chemo. It's somewhere around 30 to 40%, depending on the cohort and the center. And there's really no effective way to predict or prevent this from happening. And they usually come through the gut. It's usually a gut bug that translocates and gets into the bloodstream in most of these patients. And I said that these numbers vary a little bit, and talking to people at different centers, I've realized that there's really no standard practice for antibiotic prophylaxis and other, when do you provide neutrophil growth stimulators and things like that across centers. Everyone has sort of their own their own way of doing it. In some places, everybody gets antibiotic prophylaxis. In other places, few people get antibiotic prophylaxis. So we wanted to understand, are there risk factors that people are carrying into the hospital with them that are associated with bloodstream infection in the gut microbiome? And we first took this very simple look at the diversity in the microbiome when they walked into the hospital. And what we saw is that people who went on to get an infection had much lower diversity than when they walked into the hospital than people who did not get an infection. And these are culture-confirmed bloodstream infections. And this is basically a normalized measure of how many species do they have in their microbiome. So that was interesting. I mean, it was like night and day. Really different microbiomes walking in. Then we wondered if we could not just stratify patients, but actually predict who was going to get an infection based on their baseline microbiome. And we started with just the clinical data that we had, wondering whether maybe there are just certain factors. How recently did they have antibiotics? What's their BMI, gender? Did they have chemo previously, and how long ago? Wondering if these were simply the predictors and the microbiome was just reading out some combination of these. And in fact, none of these were good predictors of whether people would go on to get an infection. So we instead turned to some tools that we have at our disposal from our computational biology work, trying to map this raw data to a set of risk factors that could be used to predict the infection. We combine this with some machine learning approaches that we found are useful in building predictive models. And we came up with this diagnostic panel. So these are, at the top, these are the kind of potentially beneficial or protective microbes. And you can see each patient here who got a bloodstream infection. That's what BSI stands for. These people tended to have a lot less of those so-called protective bacteria than the bacteria at the bottom. These are the ones that are associated with a negative outcome in the model. And you can see that people who went on to get an infection had much more of those sort of negatively associated bacteria. And our algorithm takes the whole panel of those. You can think of it kind of like a breast cancer panel or something where you're using genes in the person's genome to predict an outcome or risk. Here we're using genes in the microbiome to predict risk. So we take this panel and combined it, and we asked the algorithm to predict the infection risk. And what we got is this. So this is what we call a leave one out cross-validation analysis. And what that means is that, say, for this particular patient, we removed that patient from the analysis, trained the model on the other 27 patients, and then asked it to predict the risk for that held out patient. So the model had not seen the answer for that patient. And what this means is that there's no information leak in the model, and it doesn't get to kind of cheat and overfit the data. And then we predict the risk for that patient, which fell here. And then we put that patient back in, take another patient out, retrain the model from scratch on the other 27 patients, and predict that held out patient. And we do that for every patient, and we get these predicted scores. So if there were no predictive association between the baseline microbiome and the risk of infection, then these scores would look like this, totally random. And in fact, when I saw this plot, I thought, that looks too good to be true. Let's do a sanity check and just scramble the labels and retry it. And that's how we got this plot on the right. So there is a strong predictive association. The if we kind of look under the hood at what microbes there are and what the panel looks like, this is basically the outcome. Low-risk patients tend to have more biodiversity. High-risk patients have less. Low-risk patients have more kind of protective bacteria that we see being associated with health in other studies. And the high-risk patients tend to have more opportunistic pathogens. And then the hypothesis is that the inflamed mucosa that's caused by chemotherapy might be protected partially by the commensal microbiome. And this is a maybe 2-week window where people are at high risk of infection. So it may be that the chemo is disrupting their microbiome, but they start off with such a strong microbiome that it's able to kind of make it through long enough to keep them from getting the infection. Now, this is still a hypothesis, so I've not shown that it's causal. It could, again, just be telling us something about the immune system. It could be telling us something else about the state of the patient that where the microbiome is just correlated with the outcome. It's not actually causal. Although it could still be used to stratify patients or as like an early warning indicator. Hey, maybe this patient you don't want to give prophylactic antibiotics to because they're walking in with such a healthy microbiome, it's probably going to increase their risk of infection. Or this patient's walking in with a severely depleted microbiome, you might want to think about giving them neutrophil growth stimulators early. And as I said, when we look under the hood at the microbes that are associated with protection, these tend to be good microbes so, from other studies. Barnsiella has been shown. So people who don't get an infection have much more Barnsiella or Barnsielaceae. And this has been shown to protect people from C. diff infections in other studies. Christensenella, this is another probably beneficial microbe. It's the most heritable microbe. There was a big twin study in the UK showed that this one is the most heritable of all the species and is actually associated with leanness. So people who inherit Christensenella tend to be more lean than people who don't. And then Fecalibacterium, again, is much higher in people who didn't go on to get an infection. This is kind of a canary in the coal mine microbe. We see it in every single disease that we've studied and even that other groups have studied. It's higher in the healthy subjects. This is one that if I had it on the table in a pill, I would eat it right now. It's a good microbe, I'm sure of it. But you can't get it. It's not available as a probiotic yet. You could order it from ATCC and grow it and take it. So actually, we should think about doing that. But it's not, we're not quite there yet. We don't have the desired probiotics or biotherapeutics. So this makes sense. I mean, just looking at this looks like a healthy microbiome. If people walked in with a lot of Barnsiella, Christensenella, Fecalibacterium, I would say, even not having done this study, that looks good. That looks good. And then we can see some of the functional signatures of risk of infection. These are some of the same things that we saw associated with chemotherapy. For example, histidine metabolism is higher in the people who don't get infection. This has been associated with protection from, it's been associated with an improved mucosal barrier. Some of these other ones, xenobiotics are higher in people who get infections. These are associated with Enterobacteriaceae growth and virulence, they tend to be. So it's clear that there's an association. Again, we don't know that it's causal, but I think there's a lot that we need to think about how should we customize treatment for patients based on their microbiome. And this is kind of the next step for the microbiome field is, okay, we can identify unhealthy microbiomes, sometimes using a correlation, like I've shown you. In some cases, we can show that it's actually causal in mouse experiments. But how do we then change the microbiome? How do we modulate it? I mentioned FMT, or fecal microbiota transplantation. I also said the word probiotic, which I know is kind of a dirty word in a clinical setting. And I think either we need to rethink what we think of when we say probiotic, because obviously the probiotic industry is this massive industry that's kind of fraudulent and very problematic. But there's been this other term, live biotherapeutic, that's been adopted for a clinical setting to say it's a treatment consisting of live microbes that are going to go in and take root or change the metabolism. They're basically clinical probiotics. So that's one option. You give people live bugs. Another option is change the food for the microbes. Try to feed the good bugs at the expense of the detrimental microbes. So that would be a dietary or prebiotic intervention. And you can also combine these. So you could give someone a set of microbes, live microbes, and package it with some of the prebiotics that those microbes need to take root. So I'm going to talk now just about some of the work we've been doing in these areas and some of the limitations, but also some of the hope, I think, that remains for future work in these two types of approaches to modulating the microbiome. So in fecal microbiota transplantation, like I said, this is really the blunt sledgehammer approach. You literally take stool from a healthy person, strain it, put it in some buffer, and administer it through an enema. And we wanted to find out if this very blunt approach actually did restore the microbiome following chemotherapy. So we did a mouse experiment where we administered 5-FU to mice, some of whom had ampicillin as sort of to simulate antibiotic prophylaxis that a lot of patients are getting, and some of whom did not. So we have either 5-FU or 5-FU plus ampicillin. And then in half of those mice, we gave them an FMT from a healthy donor mouse right after they got the chemo. And actually gave them 3 FMTs. So this is an oral gavage of mouse stool that's been homogenized and put in a buffer. And what we found was that in the mice who did not get FMT, the diversity decreased significantly after getting the chemo. So these are the mice who had these are actually the mice who had antibiotics and chemo. And you can see their diversity tends to be up here. Like I said, this is kind of the number of species, or a normalized version of that. Their diversity is up around 40 or so at the beginning, and then it goes down and stays significantly down up to a couple of weeks later. In the mice who got the FMT, it's down at first, but actually recovers by 2 weeks later. And is restored and is no longer significantly different from their diversity before chemo at baseline. So in terms of we call this alpha diversity. It's the biodiversity of the ecosystem. You can restore it with FMT. And there's been some work in this by Eric Paymer's group in patients where they're getting back their own microbiome. That actually may not be the best idea for every patient. If you believe that some people carry a very high-risk microbiome, then they may not want their own microbiome back. It may be that you want to tailor the FMT for the person depending on which microbiome they're starting with. So this is the diversity. We can see we can recover that with FMT in mice. These are mice who, this is what we call beta diversity. That's basically the ecological distance. So from their baseline sample. So this is a mouse at day, let's look at the purple ones, at day 16. And this is saying, how different is this mouse's microbiome from their baseline sample at day 1. Is it almost the same microbes, same ratio, or has it changed a lot? In this case, it's changed quite a bit. And it's significantly different from how it started. When we give the mice FMT, then we find that by day 16, they're no longer significantly changed from their baseline microbiome. So they've kind of moved away in microbiome space during the treatment, but the FMT helped them move back to a similar state, ecological state. And we can see that in the mice who don't get FMT, they lose some of these potentially beneficial microbes like Clostridium scindens. There's that Fecalibacterium that I mentioned earlier. And they're picking up microbes that often are the ones that infect patients, Enterococcus and E. coli. So they have significantly more of those by the end of 2 weeks. Whereas the mice that get FMT, they have restored C. scindens, they have restored F. prausnitzii, and they don't have a significant amount of E. coli or Enterococcus. So it seems as though FMT from a random healthy mouse can help them stay in a healthy configuration. But as I said, we may not necessarily want to give people their own microbiome back. And I think that it really is going to be probably somewhat personalized which microbiome someone needs. So to illustrate that point, I want to tell a short story about a project we did on probiotics. And the kind of motivation for this is that, as I said, most probiotics don't work. This is a picture I took at the Whole Foods in St. Paul. And it's amazing. You know, there's this like great selection of all these probiotics. They're all $30. And if you look at the back of it, it's the same microbes over and over in all these different probiotics. And most of those microbes are adapted to live in the infant gut and grow on milk. These are Lactobacillus and Bifidobacterium. They're probably good, but they don't necessarily live in the adult human gut. And sometimes they're not even viable. Sometimes the microbes that are there are not actually the microbes that are claimed on the label. So it's a very unregulated industry. Totally makes sense that it's gotten a bad name. And to kind of address this concern about probiotics that aren't the right bug, you know, they're not made for the human gut, one of our collaborators at the University of Alberta, Jens Walter, ran this interesting study where he found this probiotic that actually does engraft. And so this was called by the microbiome journalist Ed Yong, a probiotic that actually lasts. And yes, microbiome journalist is a type of journalist now. And so what they did was kind of amazing. They just grew a bug out of a healthy person's stool and cleaned it up and gave it to a bunch of people as a probiotic. Would you do that? Yeah, I mean, well, you know, if you're starting with a healthy microbiome, throwing one microbe in there is probably not going to perturb the whole system. It's like, you know, if you go into the jungle and throw in a bunch of dandelions, you know, they're just going to die. If you throw in something that's extremely invasive, maybe you've got a problem. So what happened is, but this was a Bifidobacterium strain, so they started light. You know, this is a very gentle, safe microbe to put into people. And what happened was they gave the microbes to these people, and for about half of the subjects, they saw this. While people were taking the probiotic, they could measure it in the stool. They're actually culturing it, so it was coming out alive at the other end. And then, as soon as people stopped taking it, which was after a couple of weeks, it just flatlined. Boom, it was gone. And this is very typical of probiotics. They don't engraft. They just pass right through. But then in about a third of the subjects, it did engraft. So even though they stopped taking it at 2 weeks, it was still there a month later. It was still there 2 months later, persisted up to 200 days when they stopped measuring it in these subjects. So somehow this was actually taking root. And the question that they had then, and this is why they approached my lab, was could we help them understand why it didn't engraft in some subjects? Is there a reason why people were kind of susceptible to this probiotic? So we ran similar machine learning tools, and the basic finding was that people rejected it if they already had one of those. So if you already had a very similar Bifidobacterium longum, you would reject the probiotic. Makes sense, right? The ecological niche, whatever it is that microbe might have come in and filled, was occupied by something that was already established and probably in large numbers. So here this is showing the ratio of this species in non-persisters versus persisters. So it tends to be much higher in the people who did not, in which the probiotic did not persist. But that wasn't the whole story. So there were a few other species that were associated with non-persistence, meaning rejecting the probiotic. And there were others that were associated with persistence. So if you had more Agarthella, that's the one that breaks down digoxin in some people. If you had more Agarthella, then you tended to acquire the probiotic. So maybe it plays nice with Agarthella. And in general, it's not just as simple as finding one ecological niche that's not fully occupied. It may be that probiotic, in order to take root or engraft, needs to play nice with the other microbes that are there. So I think this is just scratching the surface of what we'll see in the next 10 years or so in terms of customizing live biotherapeutics to fit what a given person needs. The other approach would be the kind of shotgun approach. So if I were going into the hospital for a high-dose chemo and I found out that my microbiome was on the low diversity end, the high-risk end, if I could design any probiotic I would just take every possible strain of every beneficial microbe I could get and take them all at the same time, hoping that some of them would stick. So that would probably be the easiest way to treat people, is rather than trying to figure out exactly which strain of Bifido longum this person needs, just give them all of them, and one of them will probably take root. The problem with that is FDA. How do you get that? Now you have to justify all of those. You have to manufacture them. You have to close their genomes. That's never going to happen. So we're sort of stuck. And actually, FDA is really struggling with how to regulate live biotherapeutics. Right now, they're letting people do FMT for free. But in terms of manufacturing drugs that are made up of microbes, it's still a very arduous process. So that's one issue, or the opportunity is there to give people custom therapeutics, and I think we will get there, but you can't buy these bugs right now. They're not produced. It's really hard and expensive to make them, and they won't necessarily work for every person. So there's some complexities there. And then the other thing I wanted to talk about is diet. And this is a little bit of a new area for my lab. It's kind of the elephant in the room in microbiome studies. If you look at every microbiome study that's been on the cover of Nature and Science in the last 5 years, the vast majority of them have no information about diet. Or a couple of them may have collected diet, but it's not clear exactly how to analyze diet microbiome interactions. And yet, diet is the major driver of microbiome composition. So we do need to do something about this. If we're going to understand how do we manipulate microbiomes, we need to understand how diet is interacting with the microbiome. Because it could be that you put all the live biotherapeutics in that you can get, but if the person's not eating the right precursors for the new microbes, they'll never take root. And then they'll never protect the person. But we really have limited methods to analyze diet microbiome interactions. So we've been starting to explore possibilities here, and we actually we've been collecting diet in quite a few studies in the lab, but we haven't known what to do about it. And I was recently lucky to meet Abby Johnson, who got a Ph.D. in nutrition here at the U, and then was actually working at Nestle, and decided to come back and do a postdoc. And she knew statistics and knew how to code, so she's joined my lab basically to try to help us crack this diet. How do we do big data mining on diet data, and how do we link it to the microbiome? So she ran this study in the spring that was kind of interesting. It was a citizen science class. So the research subjects were the students, and they, for the first few weeks of class, they collected a stool sample every day, very tiny stool sample, and they also collected their entire dietary history every single day for 17 days. So we have very dense longitudinal data now on what is the microbiome every single day, what is the diet every single day, and we're trying to understand whether the daily diet variation is actually influencing the microbiome, and even just to get a handle on how to analyze this. This is using an approach that we've developed in the lab for measuring species profiles in a very high throughput, affordable way. That's nothing fancy. We call it shallow shotgun sequencing. So we're basically saving as much money as we can by sequencing less, but using better methods to get species level information out. So we're able to do this whole study quite affordably. And it's really the first look at this kind of dense longitudinal variation at the species level in anything more than one or two people. This was actually, it was co-sponsored by the College of Biological Sciences, who picked up some of the cost, and General Mills. So what you're seeing here in the top row is the microbiome, and every column, every kind of group here is all the samples from one person. If you squint at it, what can you tell about people's microbiomes? Well, they're different. So there's some shared species. You can see this red one. A lot of people have that one. This kind of teal one, a lot of people have that one. So there's some shared species, but everybody has kind of their own fingerprint. It's maybe some of the same species, but different ratios. And they tend to keep it over time. The bars here, every column in this little batch is a different person's microbiome, and a different day from that person. And so you can see from day to day to day, they're kind of keeping the same mixture of bugs. Now, on the bottom panel here is their diet. And this is much more variable. So the daily changes in diet for these subjects was much more variable than the microbiome. And it's also very personalized. So this both made our heads spin and also got us really excited because we saw this opportunity to try to do something interesting with the data. So we started off just doing the simplest thing, something that's been done in other microbiome studies. Let's take the nutrients from people's diets and find out if it tells us what their microbiome is. So by nutrients, I mean literally, look at the nutrition label on all these foods, and we're going to look at sodium, protein, saturated fat, these sort of boring things, and ask whether your average nutrient profile predicts what your average microbiome profile is. So I'm going to So what happened is we've taken each person's average microbiome profile and made a cloud for those. So people with similar microbiomes are close to each other in the cloud. Then we took their average diet profile and made a cloud for those. And then we rotated them and stretched them until they lined up as best as possible. This is a typical analysis we do. It's called Procrustes analysis. I don't know if you know the Procrustean bed. Procrustes had this bed that he would stick people in, and if they were too short, he would stretch them out, and if they were too long, he would like chop off their legs. So it's stretching to try to make them fit as best as possible, and we can put a p-value on it. So the average nutrient profile, if your average nutrient profile predicted your average microbiome profile, then we would see that there was a significant correspondence between these two clouds. In other words, if you and I have similar diets, then we're also going to have similar microbiomes. That's what it would say. But they didn't. And we thought about this. We thought, okay, maybe that makes sense, because what's shown on the nutrition label doesn't tell you any of the complex compounds that are in the food. There are all of these complex polysaccharides. There are hundreds of different compounds when you take a bite of something that we have no idea what they are. We have no idea how cooking changes them. So we decided to look at this instead of based on these kind of standard nutrients. Let's look at the food choices for people. And I'm going to show you a similar type of plot showing the food choice network. So this is a depiction of people and their foods, their food choices. The white dots are people, and then the colored dots are the foods they're eating. And I mean literally, like, I ate black beans. That's a dot that I'm connected to. And then they're sort of positioned to be close to each other, if they're sharing foods. And what you can see from this is that it is highly individualized. So there are some foods in the middle here that everybody's eating. Black beans are probably somewhere closer to the middle, or rice. But then each person has their own pet food, not pet food, but pet foods, well, maybe it was in there, that only they ate. So we're starting to, this is really fun for us. It's like we've never looked at diet in this way. And there's some of these core foods in the middle, and there are a lot of personalized foods. And what we realized was, okay, we've got this problem right now. The nutrients don't tell us that much, but the foods are too personalized. So can we find a way to share information between related foods? And we actually found a way to do this. So Abby realized that the way that the food database was structured, we could actually organize all of the foods into a hierarchy, into a tree. And what this means is that if you eat a particular type of leafy green and I eat a particular type of leafy green, and someone else eats an Oreo cookie, we don't want to treat those as features A, B, and C that are completely independent. We want to share statistical information between the two types of leafy greens. Even though they're not exactly the same, they probably have more polysaccharides and other compounds in common than with the Oreo cookie or an egg or a chicken. And the tree, once it's in a tree like this, we have this whole suite of tools we can use from multivariate ecology. And I'm not going to go into those, but just take my word for it that once the data are tree structured, we kind of know what to do with it. And the goal is still to understand whether the diet is predicting the microbiome, and can we use this information about similar foods to better explain microbiome variation. So we've now used the tree to make a microbiome, a diet cloud, where two people are close to each other if their diets are similar. But now, if one person's eating mostly this type of salad and one person's eating mostly that type of salad, now they are going to be close to each other, even if the foods in the salad are not quite the same. And so we lined that cloud up with the microbiome, and now there's a highly significant correspondence. You can see the connections between, so the pink ends of this bar points to the microbiome sample, and the blue end points to the diet. So you can see that there's some correspondence between which people are similar in microbiome space and which people are similar in diet space. And this is actually longitudinal data. Here I'm showing you the averages, but we went into each person. And here I'm showing you where one person's microbiome was moving around in this high-dimensional microbiome space over time. So the horizontal axis is time. And you can see, so the bars are connecting where their diet was in this space and where their microbiome was in this space. So you can see their microbiome is kind of staying the same, and then it starts to change, and then it kind of stays the same in this other state. I know this is sort of abstract. I'm not telling you what the state is or which microbes are changing. But the point is, the diet is changing with the microbiome, or vice versa. So we can actually predict the trajectory of a person's microbiome over time from day to day based on the dietary changes. And we can do this for over half of the subjects. This is just a kind of introduction to something new that I think the microbiome field needs, and that I think in customizing treatments for cancer to the microbiome and customizing chemo, choice of chemo agent, dosing to the microbiome for a patient, I think we also need to be looking at people's diets. And I think it could be that you could just give people the right diet, if you can get them to eat it, during that high-risk period where they're getting the chemo, and that might be enough to reduce risk of infection. And that would be really exciting because it's very low risk, although it's much easier to get people to take a pill than it is to change their diet. So we're doing a follow-up study with 150 subjects, and these are people getting high-dose chemo for any type of bone marrow transplant or stem cell transplant, any cancer type. We're collecting stool samples before, during, and after hospitalization, and not just looking at infection, but also graft versus host disease, diarrhea. And then we're doing machine learning on this to discover microbiome biomarkers. So can we predict, based on the microbiome and how it changes either during chemo or after transplant, can we predict GVHD? Can we predict the infections? And we're also collecting complete dietary records so we can understand, are the microbial risk factors explained by the diet? And could we make dietary recommendations to shift people into a healthier microbiome state? And that might be something that could actually get into clinical practice faster than having to make a drug containing microbes in it. So that's, I've told you about what chemo does to the microbiome, how the microbiome may be related to risk of infection in chemo patients. There's a lot of other work I didn't talk about being done by other labs on the microbiome and actual efficacy of cancer treatments. That's very exciting. And then I discussed two of the ways in which we're starting to gain traction in terms of manipulating the microbiome. Either putting in new bugs, where there's some work to be done, but I think there's promise, and also just feeding the bugs better, changing diet and giving people precursors. So I'm going to stop there. I'd like to thank the people in my lab who've done this work, some of whom are here. There's Abby in the back, his work I talked about. And there's some undergraduates as well. This is us on a recent climbing outing. And the various funding agencies who funded this work, many of the collaborators who are contributing to this work. And I'd be happy to take any questions.
Dan, that was great. And, your initial setup, the idea that the reason microbiome is important is because it interacts with the host in various ways. So we have a lot more small bowel than we do large bowel. And as I understand, small bowel has a microbiome that's not identical to large bowel. Mm-hmm. And I know we do large bowel because we can sample it. Mm-hmm. I don't know if you have any thoughts about how to control for that factor, mm-hmm. especially diet, because I would have to think dietary changes both gastric and small bowel mm-hmm. microbiome.
That's a really good point, and it is a limitation. In a disease where changes are localized to the small bowel, this is present, for example, in ileal Crohn's disease, we do see shifts in the stool samples. So it's partly obscured by the rest of the signal. It's kind of a mixture problem. You're mixing some of the small bowel microbes with the large bowel microbes. And it's a problem of just needing more samples, larger studies to overcome that noise.
Yeah, I'm just, there are people who walk around with no bowel and they have a ileostomy bag. I wonder if it'd be an interesting group to study.
Yeah, I think we've thought about ileostomy bags. And I think it would be very interesting. Obviously in mouse studies or animal studies, there's a lot of work in looking at localized changes, and so I think that there's some promise there. But yeah, ileostomy bags would be a very interesting approach.
Other questions?
That really was fascinating. Thank you. So, how long does it take you to genetically profile a patient's microbiome so that you could use it, for example, to make a clinical prediction? Is that weeks or days or afternoon? Or how does that?
Do you mean how many samples do we need, or what's the actual turnaround time?
Well, both. I mean,
Yeah.
what do you have to go through to say, oh, you have this
Right.
microbiome?
Having one snapshot from a person is pretty good.
Okay.
Two, if you got day 1 and day 2, or day 1 and then a week later, you're sort of better able to triangulate around some of that daily variation.
Okay.
But so having a couple of snapshots is a little bit better. In a study, I would get like 1 or 2 or 3 daily samples if I could.
So what would you need then if you wanted to use this clinically?
And then what do you do? Yeah. So right now, it's tough to do the sequencing. You can do it in a day. You basically have to extract the DNA, prepare sequencing libraries, and by now it's like 4 o'clock, and then you can run it on a sequencing run that's pretty quick and have it back the next morning. Okay. But you have to have a full-time person, and you might spend a lot of money because if you don't have enough samples to fill up the sequencing run, you might be paying like $1,000 a sample.
Sure.
So it's doable. It's doable in 24 hours with a really specialized operation.
Okay.
And I think we're going to see big shifts toward DNA sequencing as an approach for evaluating infections, like sequencing blood, sequencing stool, rather than, we can already do it faster than, for example, having to try to grow the bugs that are infecting someone and wait like 5 days, and meanwhile the patient's dying,
Right.
to find out which antibiotics to give them.
Right.
So it's doable. It's going to take, I think, a really focused commercial effort to make it, to get it into the clinic with a 24-hour turnaround time, but it's going to happen.
Okay. And wait, one more quick one, sorry. So it must depend on the type of chemotherapy that you're getting too, right? So docetaxel, I would assume, I don't know what to assume. Would it have the same impact as 5-FU or?
We don't know. Yeah, we've really only looked at 5-FU. Chemo tends to target rapidly growing cells, which is the gut lining. And so that's the hypothesis, that it goes in there, disrupts the gut mucosa, causes inflammation and a leakier gut, and then the bugs shift in response and leak through into the blood. But we really haven't done any systematic study of other types of chemo agents.
Okay, thank you for this interesting topic. Do you think the changes in the microbiome depend on the type of chemotherapy?
Yes, I think they will. And I think there's going to be a pairing between the type of agent and the microbiome. So irinotecan, for example, is actually reactivated by the microbiome after it goes through the liver and causes toxicity and diarrhea that can affect dosing. So it's absolutely going to have an effect on the change in the microbiome. And I think there may even be cases where there's an interaction term there. So basically, like depending on whether your microbes metabolize or interact with that drug, you will have a different shift in the microbiome.
Any other questions? All right. Thank you very much.
Thank you.